So, good day everyone. So, for today's video, we're going to discuss the concentration of phosphate in freshwater algae. But first, what is phosphorus? So, phosphorus, like nitrogen, is a critical nutrient required for all life. The most common form of phosphorus used by biological organisms is phosphate or PO4, which plays major roles in the formation of DNA, cellular energy, and cell membranes, and or plant cell walls. The phosphorus is a common ingredient in commercial fertilizers. And why is it important to evaluate the concentration of phosphorus? The high concentrations of phosphorus may result from poor agricultural practices, runoff from urban areas and lawns, leaking septic systems or discharges from the sewage treatment plants. Too much phosphorus can cause increased growth of algae, which can result in decreased levels of dissolved oxygen, a process called eutrophication. The high levels of phosphorus can also lead to algae blooms that produce algotoxins, which can be harmful to human and animal health. So this is what it looks like when there is a higher concentration of phosphate in fresh water. So as you can see, the water turned into a green color. To determine the concentration of phosphate, conducting a spectrophotometric analysis using the colorimetric method is the perfect method for this analysis. The apparatus used are the UV or spectrophotometer, volumetric flask, beakers, and pipette. And the reagent used are the water sample, the vanillate molybdate reagent, and the standard phosphate solution. Procedure. First, shake the water sample to ensure thorough mixing, then transfer a 25 ml aliquot to each of two 50 ml volumetric flasks. Second, add 10 ml of the vanillate molybdate reagent to one of these flasks. The second acts as a sample blank and dilute each to the mark with the ionized water. Third, shake to mix. Fourth, repeat 1 to 3 for the other water samples. Fifth, leave all prepared solutions, including the calibration standards, to stand for at least 10 minutes for the color to develop fully before taking absorbance readings. 6. Measure the absorbance of each standard sample in blank at 470 nanometer. 7. Plot a calibration graph of absorbance versus phosphorus concentration in grams per milliliter for the standard solutions. And last, use this graph to determine the phosphate concentration in each water sample. Remember to allow for the dilution of the sample in your calculation and subtract the sample blank reading from the corresponding sample. And calculate the concentration using the Berlambert's law, which is the concentration is equal to absorbance divided by the molar absorption coefficient times the path of length. And according to the article, the peak concentration and environment directly affected the growth of algae, including the population density, specific growth rate, photosynthetic pigment, and food quality. Consequently, it was concluded that the peak content not only affected the growth of primary producer directly, but also affected the subsequent consumers indirectly via alteration in the stoichiometric food quality. This study may improve our understanding of the underlying role of phosphorus in affecting aquatic food chain and complex natural environment.